Hello friends, welcome to my channel Pharmagyan. Friends, in this channel you can learn about pharmaceutical guidelines, pharmaceutical industrial practices, processes, documentation practices, and many more about pharmaceutical industries. Friends, please don't forget to like, subscribe and share my channel Pharmagyan, and press bell icon given in bottom right corner to get latest updates directly in your notification panel when I upload new videos. So next we will discuss about the video.
Part 7 of 8. Experiments possible acceptance criteria. Accuracy. Perform a minimum of 9 determinations over a minimum of 3 concentration levels covering the specific range. For example, 3 concentrations/3 replicates each of the total analytical procedure. Determine analyte with respect to the total amount of residue in the sample, for example weight/weight. Accuracy as % recovery or 90.00 to 110.00%. Difference between the mean and the accepted true value. 10.00%, P equals 95%. Confidence intervals. Precision. Investigate using homogeneous, authentic samples or, if not possible, artificially prepared samples. Perform a minimum of 9 determinations covering the specified range for the procedure, for example 3 concentrations/3 replicates each, or a minimum of 6 determinations at 100% of the test concentration. Repeatability, intra-assay precision. Establish precision under the same operating conditions over a short interval of time. Report. Standard deviation, interdependent with SREL. See SREL. Overall relative standard deviation over the whole range of the method. 10.00%. Relative standard deviation within one concentration level. 20.00%. Table 2, validation requirements, continued, confidence interval. Intermediate precision may include robustness, ruggedness. Establish precision on different days, for different analysts, on different equipment and after variation of method parameters, equals robustness, for example stability of solutions, variations of pH, of mobile phase composition, of flow rate, of temperature, of columns etc. It is not necessary to study these effects individually. Experimental design, matrix, may be applied. Report. Standard deviation, interdependent with relative standard deviation. See SREL. Relative standard deviation. 3 times SREL from repeatability or 10% whichever is greater. Confidence interval. Specificity. Demonstrate the discrimination of the analyte in the presence of the other impurities. Test samples containing the analyte and other impurities. Obtain positive and correct results for the analyte. Specify acceptable deviation. Test samples without the analyte. Negative results. For chromatographic procedures, use representative chromatograms to document specificity. Label individual components appropriately. Specify acceptable resolution of peaks. Experiments possible acceptance criteria. Linearity. Measure a minimum of 5 concentrations across the range of the procedure. Dilute standard stock solution or prepare synthetic mixtures. Plot the signals as function of concentration. Evaluate the plot. Visually. Linear. Statistically, for example regression line by the method of least squares. Correlation coefficient, y-intercept confidence band, P equals 95%, contains 0. Slope of the regression line. Residual sum of squares. Range. Confirm that the analytical procedure provides an acceptable degree of linearity, accuracy and precision within or at the extremes of the specified range. Minimum specified ranges. From the reporting level to 120% of per max. The reporting level for cleaning validation reasonably will be the look. However, the reporting level must be below per min and should be below or at 80% of per min. From look or 80% of per min to 120% of per max.